Hello again, Psych 370 students, and welcome to your first video lecture for week 8. Last week, we focused on the first part of chapter 5 of the textbook, which covered the basic principles of operant conditioning and some of the variables that affect reinforcement. So this week, we're going to finish up that chapter by reading about the physiological basis of reinforcement and also about some of the theories that have been proposed to try to explain what exactly a reinforcer is and why so-called avoidance behaviors get reinforced. So that's what most of the second half of the chapter is about. And so as usual, in this week's video lectures, I'll be talking about that stuff. I'll try to clarify some of the concepts and some of the theories that you'll be reading about. And in this first video lecture, what I'm going to focus on is the physiological basis of reinforcement. So what's going on in the brain, in other words? Or to put it the way your textbook puts it, what are the neuromechanics of reinforcement? Well, guys, interestingly enough, the first major breakthrough in this area of research was actually made by accident. So there were these two researchers, James Olds and Peter Milner were their names. Olds was actually an assistant professor at McGill University, and Milner was a graduate student there. And they thought that if they stimulated a brain region called the reticular formation in rats, then that might facilitate maze learning in the rats. So the reticular formation is involved in attention. Okay, so what Olds and Milner were thinking was that if they stimulated the reticular formation, then that might increase how attentive these rats were to their environment, and that might allow them to learn this maze task more quickly. So that was their hypothesis. But when they implanted their electrodes in these rats' brains, Olds and Milner missed their target. So instead of stimulating the reticular formation like they'd intended, they actually wound up stimulating a part of the brain called the septum, the septal region, which is located about right here, and which, as you can see, is very close to the hypothalamus. In fact, there are lots of connections between the septum and the hypothalamus. The septum has axons that project to the hypothalamus, and so the hypothalamus wound up getting activated by this brain stimulation that the rats were receiving. And that'll be important later. So remember that increased activity in the hypothalamus was being caused by this electrical brain stimulation that Olds and Milner were applying. Now, before they actually conducted the study, put the rats in the maze, Olds and Milner first wanted to make sure that this brain stimulation wouldn't be aversive for the rats, because that would obviously interfere with their ability to learn the maze task. So they tried it out first. And when they did, they found that this brain stimulation actually seemed to be reinforcing for the rats. So for example, they had these rats in a sort of tabletop enclosure like this, okay? So whenever a rat entered a particular corner of it, that's when it would have that electrical stimulation applied to its brain. And not only did the rats not act like that brain stimulation was aversive, it was quite the opposite. They actually strongly preferred to remain in or return to that part of the enclosure. So in other words, they developed what we would call a conditioned place preference. Okay, so they associated this part of the enclosure with that brain stimulation, and so they acted like they preferred that place. They had been conditioned to prefer that place, conditioned place preference. And so to put it like Olds and Milner put it, it seemed as though these rats kept coming back for more. They kept returning to that location. Furthermore, Olds and Milner also reported a study where they made a lever available to the rats, and they made electrical stimulation of the brain contingent on pressing that lever. So in other words, they rigged up a situation where these rats could self-stimulate their own brains. The rats could cause that electrical brain stimulation to occur by pressing the lever. And what they found when they did this was that those rats would press that lever thousands of times per hour. Okay. So that brain stimulation acted as a very powerful reinforcer, right? It reinforced the rat's behavior. In fact, it was even more reinforcing than food. So when these rats had a choice between one lever that produced food and another one that produced electrical brain stimulation, they chose the one that produced the brain stimulation, even though they didn't have any other way to access food. So many rats starved to death as a result. And if they were allowed to press that lever continuously, they'd do so for as long as 48 hours straight until they finally collapsed. So, when pressing that lever produced electrical brain stimulation as a consequence, that behavior, pressing the lever, got much stronger. Again, it got reinforced. But of course, it's not the case that stimulating just any area of the brain would have done the same thing. So remember, Olds and Milner got the results they got with those rats because they placed their electrodes in or near the hypothalamus, which, as I've said on the slide here, is an important part of what's sometimes called the reward system or the reward pathway in the brain. And so these are some of the brain areas that animals will basically work the hardest to stimulate. They all have connections between them. This medial forebrain bundle here is a bundle of axons that connects them with each other. And guys, what they all have in common is that when you stimulate them, it causes the release of dopamine. It causes an increase in the levels of a neurotransmitter called dopamine, especially in this region right here, which is called the nucleus accumbens. And so, not surprisingly, other reinforcers do that too. For example, addictive drugs, sexual activity, sugar, music, gambling, video games, all those things are associated with increased dopamine release in the nucleus accumbens. And it's also worth pointing out here that if you give an animal a drug that blocks dopamine activity in the nucleus accumbens, then guys, that reduces the reinforcing effect of electrical brain stimulation. In other words, animals won't work as hard to stimulate their brains if doing so doesn't cause dopamine levels to spike in the nucleus accumbens. And so that's probably the most basic thing to know about the physiological basis of reinforcement. Whatever specific reinforcer you're talking about, increased dopamine release in the nucleus accumbens is the common thread that seems to tie all of them together. And guys, just to dig a little deeper into this, there's also a relationship between, on the one hand, how unexpected a reinforcer is, and on the other hand, how much dopamine release that reinforcer tends to cause. So the more unexpected the reinforcer is, the more it surprises the animal, in other words, the bigger the increase in dopamine that it tends to cause. So, for example, let's say that we've got a monkey who's learning to perform a simple task like pressing a button in its cage. And whenever it presses that button, or maybe whenever it presses that button a certain number of times, we reward it by giving it some kind of reinforcer, a grape, let's say. So the monkey is learning to press this button for grapes. So pressing the button is the behavior here, and grapes are the reinforcers for that behavior. Well, the first time the grape occurs as a consequence, it's going to be pretty surprising to the monkey, right? I mean, it doesn't know yet about this relationship between pressing the button and receiving grapes. So that first grape, that first reinforcer, is going to be very unexpected. And so as a result, the monkey is going to experience a pretty substantial increase in dopamine release from that reinforcer. However, as the training progresses, as the animal comes to learn about this relationship between pressing the button and getting the grapes, those grapes are going to start to become a more and more expected consequence of pressing the button, right? The monkey's going to come to expect to get grapes after it presses the button, and therefore those later reinforcers, those later grapes, are going to trigger less and less dopamine release in the monkey's brain because now the monkey isn't as surprised by their occurrence. So there is this relationship between how surprising a reinforcer is and how much dopamine release it tends to elicit in the brain. And guys, the reason that's interesting is that it might help explain the shape of the learning curve. Now, the way your textbook describes that shape is to say that it is decelerating. It has a decelerating shape. So it's usually during the first few trials that you're going to see the biggest gains in learning, right? And then on the later trials, that rate of learning decelerates. It slows down. So there isn't as much of a change in behavior on the later trials as there was in the earlier trials. Well, one reason that might happen is because on the earlier trials, the reinforcer is very surprising, right? So on those early trials, the reinforcer would have triggered a bigger increase in dopamine levels. And then on the later trials, the reinforcer is less surprising. So there's a smaller increase in dopamine now. And again, there's also a smaller change in behavior on those trials. So, guys, the idea here is very similar to what we talked about a few weeks ago with the Rescorla-Wagner model. Remember, the more surprising an event is, the more unexpected an event's occurrence is, the more you seem to learn as a result of that event. And we talked about that before with regard to classical conditioning and how surprising the unconditioned stimulus is. But the same idea could be applied to operant conditioning and how surprising the reinforcer is. More surprising reinforcers are associated with bigger increases in dopamine release. And again, that could help explain why we seem to learn the most when the consequence that we experienced was unexpected. Okay, well, that's all I have to say about the physiological basis of reinforcement, the neuromechanics of reinforcement. So please let me know if you have any questions about this stuff, and I hope you have a great day. Take care.